So welcome to the Crop Protection Network Spring Webinar Series hosted by the Southern IPM Center with funding from USDA National Institute of Food and Agriculture Crop Protection and Pest Management Regional Coordination Program. We are one of 4 regional IPM centers supported by USDA NIFA, and we have been charged to coordinate IPM across the region. I'm Kayla Watson, the Communication Director for the Southern IPM Center. And we are very proud to be partners with this series. It's the second presentation in the spring series. And today we've got speaker Dr. Trey Price, an extension plant pathologist at the LSU AgCenter. And Trey is going to be talking with us about soybean taproot decline, diagnosis, and management. So we're really looking forward to this today. Thanks for doing this, Trey.
Yeah, thanks for the invite, Kayla. As Kayla mentioned, I'm Trey Price. I'm an extension and research pathologist working out of the LSU AgCenter. I work on, at this point, 8 different crops in the state, so it's never a dull moment. And coming to you now from the Rice Research Station in Rain, Louisiana, actually. So I wanted to talk a little bit about soybean taproot decline today. It's a relatively newly discovered soybean disease. We've been having trouble with it in Louisiana for, I'd say, at least 15 years. It was only recently that we figured out the pathogen that was involved, but we've been working on it for quite some time. Now, the first thing that farmers notice when we have a field that has a problem with taproot decline is during pod fill. Right around, between R5 and R6, farmers driving down the turn row and they notice the beans there's some yellow spots in the field essentially, and they know it's too early for that to happen. So that's when we get the call, and you'll have interveinal chlorosis. If you walk out to those yellow spots, you'll have that classic symptom of interveinal chlorosis and necrosis. Now, one important thing to note here, when you have that symptom type, in my neck of the woods, you need to dig deeper because it can be a number of different issues. I've got a list here. The most popular one I would think would be sudden death syndrome. It's, I can't tell you how many calls I've had where people thought it was sudden death syndrome and it turned out to be taproot decline. As a matter of fact, in Louisiana, sudden death syndrome is exceedingly rare. But there are a number of different problems. If you have this symptom type in your field, you definitely need to dig deeper, and you need to start lower in the canopy. You need to look at the roots and the crown of the plants. So that's what we do in these situations where we have these field calls. You go out and you see the yellowing, the interveinal chlorosis and necrosis, and you part the canopy. You look down at the ground, and more often than not, there in the top left, you'll find soybean plant or multiple soybean plants that have died earlier in the season. And what happens is the neighboring plants, the infection levels weren't high enough to kill the plants, so their neighbors covered them up and the farmer didn't notice, especially during vegetative stages. This taproot decline can go unnoticed pretty quickly in vegetative stages. And in fact, there's a picture on the right here where we caught a little soybean plant there in the process of dying. So, and its neighbors showing some foliar symptoms, but the other neighbors along the row are going to quickly cover that soybean plant up and nobody's going to notice. If you dig that dead plant up, or if you dig a plant showing foliar symptoms up, you'll have a blackened root there on the bottom left. That's not really a great picture, but I wanted to show a picture with a blackened root next to soybean debris. That's pretty key. We generally see a lot of high debris situations. We see high taproot decline incidence and severity. Even earlier, even VC to V1 stage soybeans can be affected by taproot decline. I also suspect that it's seed rot as well. The plant here is, you have a V1 plant there on the left, and the leaves there, you see that mild interveinal chlorosis, and the what do they call those? Not cotyledons, but unifoliates. Sorry about that. They have a they have a unique symptom type on them. And if you look there, you can kind of see the interveinal chlorosis, but there's more of a mottling on those leaves for whatever reason. You know, maybe that'll end up being a clue for us making some other discovery on this particular disease system. But you dig those plants up again, and you'll have that diagnostic black root there. Another thing that we encounter in the field, particularly after periods of high moisture, rainfall, long dew periods, high debris situations, it doesn't matter if it's soybean debris, corn debris, whatever, cotton debris, previous crop, we'll see what's known as the reproductive structure of the fungus. They're called stromata, but we have a number of creative sayings, you know, dead man's fingers, devil horns. And my favorite came a couple seasons ago from one of my favorite consultants. He called them fuzzy fingers. So I got a kick out of that day. But recently we discovered that reproductive structure is produced by Xylaria necrophora. We have a graduate student in Baton Rouge that I'm co-advising along with Dr. Vincent Doyle. Name's Teddy Garcia Aroca, and he's done a really nice job of characterizing this pathogen genetically. He's gone all over the southern United States. You can see all those waypoints here. Collected many samples of Xylaria necrophora for his research. He chose, you know, he selected a subsample of those isolates and compared those with other herbaria samples. You know, over the years, people will collect interesting fungi and they'll store them in a herbaria, and they're usually long forgotten. But turns out Teddy found some that were very useful. He also used isolates from southern Louisiana, southern Florida, and a Caribbean island, Martinique. I don't expect you to be able to read this. This is just a genetic characterization there, the phylogenetic tree. If we zoom in a little bit, Xylaria necrophora is part of the Xylaria arbuscula species complex. It's most closely related to Xylaria striata. Teddy compared all of our isolates to one from China that came from rice, the one from Martinique that was on decaying organic matter. Dr. Allen out of the Mississippi State actually provided some samples off of cotton, Gossypium hirsutum there, several samples off of cotton. And I need to stress that was not infected cotton, that was on dead cotton debris. If you look at some of these samples that came from southern Florida, they're nearly 100 years old. So that particular fungus has been in the United States for almost 100 years. So the other isolates came, or other herbaria samples were from gardenia and sugarcane, which is interesting to note because we grow a good bit of sugarcane here in the southern part of Louisiana. So what is Xylaria? It's a fungal species, and they're known as saprophytes or wood rotters. They basically consume dead organic material. They are known as endophytes, which means they live within plants without causing disease. But there are a few pathogens in the group. One there on apple, Xylaria molly, and Xylaria acuta on cherry. Also Xylaria arbuscula on sugarcane. So for some reason, Xylaria necrophora transitioned apparently from an endophyte to a pathogen of soybean. Exactly why, we don't know. That's just the bottom line right now. We don't know yet, but we do know this organism causes disease in soybean in the southern United States. That's just an excerpt from Teddy Garcia's work, his excellent PhD research work. He's done a really good job characterizing this organism, and we're really proud of him for that. And I've got to take just a few minutes to brag on an excellent graduate student. So Teddy's future is bright. He's currently working on, in addition to his genetic characterization, we suspect that a phytotoxin that's produced by the fungus causes that foliar symptoms, very similar to sudden death syndrome. This is a lab experiment that Teddy did, looking at culture filtrates and with 3 isolates of Xylaria, and you can see there's definitely some symptomology going on there from those culture field traits. And they had a Colatotricum species and a non-treated control that showed no symptoms or phytotoxicity. Now, the importance of taproot decline is pretty important, and I can speak for Northeast Louisiana. I also suspect that Southeast Arkansas or Eastern Arkansas and probably the Mississippi Delta would say that it's a serious problem. Dr. Spurlock out of the University of Arkansas, 2018, got a good picture of the worst field he's ever seen here, and you can see that interveinal chlorosis. And you can imagine as the season wore on, it got worse. I saw a field similar to this last year in Northeast Louisiana. Tom Allen, that picture likely came from the Delta there in Mississippi across the river. That's a little further along in the season, but you can see how the plants here have died prematurely. As far as actual yield losses, I can conservatively say that in a field that's really bad, you're looking at probably 25%, but that's just kind of shooting from the hip and with a gut instinct. But Dr. Allen, Dr. Spurlock got together and did some aerial imagery and correlated those with yield maps. And if you look on the right here, you can see all these red areas. Well, those were the areas that were worst affected by taproot decline. If you look at the yield there, you have the red and orange colors, you're looking at about 30 bushels to the acre. So, and you can see the potential here, the yield potential here was pretty good. In the Delta, we can grow soybeans anywhere from 70 to 90 bushels is pretty common. When we do ring the bell around 100 bushels to the acre, every year some farmer does it. So significant yield losses there. In Northeast Louisiana, we just went on farms and actually did counts and hand harvesting. We counted disease incidence and severity and also plant death. I went out and hand harvested soybeans and got some estimates in a bunch of different locations. And a lot of variability in the data, depending on the field, because you have different situations in a lot of fields. But we found that as incidence increases, the percent yield loss increases, which makes sense. That's the case with most diseases. Some of the worst fields were 40%. Now that was 40% in the spot that we happened to sample. So it might not have been 40% field wide, but you can see how it can get pretty severe. Disease loss estimates from the Southern Soybean Disease Workers, we estimated 1.4 million bushels from those 4 states during 2019. And the preliminary data, their losses supported there or reported there from those 6 states. So if you're worried about taproot decline and you want to know where to go find it, or if you think it may be a concern on your farm, the first fields you need to look in are the ones that you've had soybeans in for more than one season. The worst fields that you're going to see taproot declining are those that are in soybean monoculture. And the further it goes along, 3 to 4 year soybean, it seems like the worse it can get if you've got a really susceptible variety out there. Reduced tillage situations. This is a debris-borne disease. It's if you have any debris accumulation or soils that where debris doesn't break down very quickly, this is going to be a problem because that pathogen will live on that soybean debris. You can take that soybean stalk right there on the left of the screen, surface sterilize it, split it, and isolate that organism with a needle, and put it directly on the media, and you'll have an isolate. So it's really easy to isolate. The other thing that we see are GPS tractors. Everybody's got a GPS now, and you're placing that seed back in the same spot every year. If you have your tractor, you have your field, and you drop the planter, that seed's going right next to that debris from the previous season, and that the pathogen is coming off that debris and infecting that plant. Also, susceptible varieties. We do see differences in varietal susceptibility, and I'll share a little more on that right here. Myra Purvis, research associate, and she was a master's student in my program. We took all of the soybean varieties that were in our official variety trials, and we did a greenhouse screening, and we saw differences among the varieties as far as susceptibilities, and some appear to be resistant. The problem with this type of screening is it takes a long time. It took us months to do this over the winter. It basically took the entire winter because it takes 3 to 4 weeks to get symptoms in the greenhouse where you can do some ratings. So it's really not a feasible way to continue on. The other thing to mention is the variety turnover in the industry is incredible. We can't keep up with the industry doing this kind of screening in the greenhouse. So in 2020, we decided just to install a copy of our official variety trial for the state of Louisiana at the Macon Ridge Station in a field that has natural infestation of Xylaria. But just to hedge our bet, I inoculated the trial as well. So we had 2 row plots, and row 1 was inoculated, row 2 was not. And I went through and rated this trial multiple times, and we got really nice symptom development from our inoculation. You can see the interveinal chlorosis. That picture doesn't show any dead plants, but there were undoubtedly dead plants in that plot or in that row. But what we've decided to do is just to provide this information on an annual basis, like we would any other disease ratings. This is an example of some group 3 to 4.4 soybean with several different diseases rated here and other attributes rated. But if on the far right, there's our taproot decline column. If a grower is concerned about taproot decline, they can go here and get this on an annual basis from now on, which is a good resource. I also tell growers if you've had beans in a field for 3 years plus, 2 or 3 years plus, and you're going back with soybeans on that 3rd or 4th year, and you had a variety, you've got some varieties that are performing well in those situations, and it hasn't been a problem, it's likely that variety is resistant, so, or at least tolerant if you haven't had a major problem after 2 or 3 years. The other thing we get a lot of questions on are seed treatments and in-furrow fungicide options. We're working on recommendations for that. We still need some more data, but this is an example of the type of trial that we've been conducted. I need to put a disclaimer on here. A lot of this is not labeled. We can get away with this on a research station for the cases of research, but double check your labels because I can tell you that a lot of these treatments aren't labeled. Dr. Allen, he didn't see anything really exciting when it comes to seed treatments. None of the seed treatments really perform better than the non-treated control in this trial. But the in-furrow applications, we had some activity with Circadus, and that has the STHI active of Preaxer. We use that product in rice, but also interestingly enough, Ridamil, which is not it's surprising to me that active works on that particular fungus, but we're going to continue to look at that one. Headline in-furrow has probably been consistent, the most consistent over the years. Looking at this the past couple seasons, Top Guard Terra worked, but you have to be careful with that. That product is not labeled in soybean, and the active in that material will actually hurt your soybeans worse than taproot decline will. So there may be an application method that FMC can change that may work. We'll just have to see. The other thing that we picked up was an older chemistry, thiophanate methyl, used as topsin for a long time. Looks promising. I don't think it's labeled for in-furrow use, but because it looks so good, I think I'm going to continue to look at it. I want to do a rate study on it, or perhaps maybe a seed treatment study. Similar results for me last year at Macon Ridge. We saw activity with actually a stamina seed treatment, which was encouraging. Again, activity with Circadus, with the headline and the topsin. So we'll continue to do this in the future. And we inoculated this trial, so you can see the inoculum worked. There's my yield line. it's always green, right? So that's the non-inoculated yields here, between 40 and 45 bushels. So we knocked it back to around the 30 bushel level with that inoculum. And you can see where we had the best disease control, we had the highest yields there in the inoculated plots. We're going to continue to work on that. Hopefully we can iron out some solutions for some of our growers, particularly with soybean prices looking favorable for the time being. It's definitely an interest in seed treatments for sure because of the convenience. A lot of people don't want to deal with their in-furrow fungicide applications. Tillage operations, with conservation tillage, most people practicing conservation tillage, a lot of farmers don't want to till, but even a minimum tillage operation, something to disturb that debris or encourage decomposition of that debris, we see reduced taproot decline incidence. Actually, I've been looking at this for quite some time. In 2011, I had a tillage trial for Cercospora blight. Well, taproot decline showed up in that trial, and I just happened to rate it. It was, even though we didn't know what was causing it at that time, we were calling it misdiagnosing as black root rot, or we even called it the mystery disease for a long time because we didn't know exactly what it was. But any tillage may help. Also, crop rotation. I've seen plenty of anecdotal evidence in the fields, out talking to farmers, asking about their operations. This particular data came from a center pivot, a field that was split in half. It had a center pivot, and the previous crop was corn on the one half and soybean on the other half. Well, where the previous crop was corn, we had significantly lower taproot decline incidence. So that's a pretty strong indicator. Also, and I apologize, I haven't updated this slide with the 2020 data, but we have a rotation trial, long-term trial at Macon Ridge Research Station, and we see with any rotation, with the 1-to-1 rotation of corn, grain, sorghum, or cotton, we see lower taproot decline incidence, and we also see higher yields. That could be from less disease, less taproot decline, or it could just be the agronomic benefits of rotation. Rotation in general is a really good practice. So to take home, whoops, I'll skip the one there. It's taproot decline is a serious problem for us, particularly in the northeast part of the state in high yield situations in these rich alluvial soils off the Mississippi River, and even further west along the Macon Ridge. We've had some significant problems with it, and it continues to be an annual issue. Besides southern root knot nematode, I would say it's our number one. The soilborne problem, I think it's more debris borne, but the debris is in the soil. So in our current distribution in the United States, we don't know. If you look at the distribution that Teddy worked on, it indicates that it may be wider than that. So, it could reach into other states, and I think diagnosis is key on finding this. It would be nice to know the extent of the distribution in the U.S. for sure. We do have commercial varieties. We are working on getting that information to that have some sort of resistance, and we're working on getting that information to our growers on an annual basis. We plan to cooperate with the University of Arkansas and Mississippi State, and just try to do one location where we're screening these varieties in the field, just to simplify things, because all the varieties are in each given state, and even across locations may not be the same. So you end up having to, there are a lot of redundancies in the South as far as what varieties are in LSU's OVT versus University of Arkansas's OVT. But there are some differences in varieties. So we're going to try to work all that out and have one big trial. Continue to look at fungicide efficacy, particularly in-furrow fungicides. Those are looking good. And again, seed treatments, we're always wanting to seed treatments a lot more convenient. Minimum tillage or rotation will, that's a solid recommendation right now for us. I think we've got a good bit of evidence to back that up. But there are a lot of unknowns about this particular pathosystem, and the research going on is fun. I'm looking forward to seeing what the other universities are going to come out with in the near future, as well as our group. There's anytime you're working on something new, it's pretty exciting, and everything you do is new, so it's fun. We've got a good group of collaborators. I apologize if I left anybody off this slide. This slide's a little dated, but we've got a good group in the Mid-South that we're all working on, and we're all communicating. So, and collaborating, we're a lot more efficient if we do that. And last but not least, I definitely want to thank the growers and the various commodity boards. We can't do what we do without support from you. And the goal of my program has always been to come up with applied solutions where I can help growers, bottom line, and that's what we're here for. And we're thankful for your support. We pay good people to do good work to solve problems for you. So with that, I'll be happy to take any questions. And it looks like there are quite a few here on the Q&A.
That's right. So everyone, I'm Kirsten Wise. I'm an extension plant pathologist with the University of Kentucky. And thank you, Trey. I think that was really good information about this newer disease, especially for people who may not have seen this before. And we'll go through the Q&A, but I want to start by saying that I got to see this disease for the first time in 2019 when I went down and visited Tom Allen in Mississippi. And I was really struck by the fact that if Tom hadn't told me it was taproot decline, I would have totally thought it was sudden death syndrome or something else. So how often do you think taproot decline gets misdiagnosed?
It could be a lot. I know in Louisiana, it got diagnosed a lot. Every time, I've been on just tons and tons of field calls. I've got sudden death syndrome, and you go to the field and it's taproot decline, and you teach them how to, teach the farmers what to look for and how to identify it. But I guess out of sheer repetition, it's gotten easier for us to identify it, and we know what to cue in on with our eyes. It was just an observation on I put the fuzzy fingers or the dead man's finger slide up. That's just out of pure repetition. Just go into the field and you're looking at the ground, you're like, what are those things? And then if you notice they're kind of coming up out of the row, out of the, where the soybean debris is in the plants, and then a light bulb goes off. So, yeah. And with sudden death syndrome, in my experience, the foliar symptoms are slightly different. With taproot decline, it can be a little more vague, and the pattern is tighter, if that makes sense. You can see more of the leaf veins with taproot decline than you can with sudden death syndrome. So to answer your question, yes, I think it gets misdiagnosed a lot.
Well, I really like this fuzzy fingers term. I think I'm gonna adopt that because I do like that. So we're gonna take a question from the Q&A. So this one is that Xylaria likes debris. And so do you think it can grow and reproduce on debris and build up inoculum over the years? And could you comment on what types of debris would be more favorable, or are there other hosts that you think Xylaria might be endophytes?
Yeah, I can comment on the debris. More often than not, I can readily isolate the pathogen from soybean debris from previous years. So if you have that, if the fungus has already infected the plant, that plant gets chewed up through the combine. You're left there with that little stub of a soybean plant, just a dead stalk. Well, that fungus is going to stay there. It's happy there. And it may move to other pieces of dead debris, and certainly, it may colonize any other kind of dead material. But that soybean debris can build up over a period of time, and in my experience, it gets to a point where you kind of build up and it just kind of explodes, and you have a major issue with disease in a given year. Okay. As far as alternative hosts, we don't know yet.
Yep. So what about resistance screening? So you talked about your greenhouse trial results. Do you think that translates into seeing similar things in the field? Would that hold up in the field?
We confirmed it with some field trials while we were doing the greenhouse work, and most of the time it did line up. I think it's better to do it in the field.
Okay.
Because with the greenhouse, you just have the fungus, you just have the pathogen, you have the growth media, the potting soil, and you have the plant. Well, in the field, you've got all kinds of other things going on. So I think it kind of, it mimics real life a little better in the field, in my opinion.
Right. And also, I had one of this question too. We got a question from the Q&A, but I also had this question about the in-furrow treatments, especially compared to your seed treatments. And some of those in-furrow products looked pretty good. So what are your thoughts on why the in-furrow treatments might be working better than the seed treatments?
Coverage. You can only get so much fungicide on a seed. If you can envision a little bitty seed with a coat of fungicide on it, and then with an in-furrow application, you can do whatever gallons per acre you can handle. Generally people will do 5, but I've seen people do 10. And you're spraying a 2 or 3 inch band right through the furrow, so you're actually covering more than the seed. You're covering the potentially infectious debris that's there, or in my case, the inoculum that I put there.
So do you think you'd see different results if you were using like 2x2 or some other method to apply that product in furrow?
It's possible. It probably depends on the product and how systemic it would be. I haven't tried that. I've actually ordered some 2x2 equipment.
Okay.
Because I know that's a very popular method for getting things in, next to the furrow. So I'm definitely looking at that. They're on back order though, so they're popular when they're on back order.
That's right. Yeah, we get a lot of questions about the 2x2 too.
Yeah.
So another question about the symptoms. So do you only see the mottling symptom in the unifoliates, or can you also see that in the upper leaves along with the interveinal chlorosis?
In my experience, the unifoliates just look different, and I don't know why.
Okay.
I think it's a clue, but I don't know. I don't know what clue. I don't know what it's going to end up. I don't know what we're going to end up discovering. Why there's a difference there.
Right.
But usually in the trifoliates, it's just interveinal chlorosis. For whatever reason, those unifoliates just look different.
Yeah. And do you think any of that could be environment? Because one of our other questions that we've often had is how much environment and some of the environmental conditions could affect taproot decline.
Yeah, the environment affects everything. I generally see the worst, and this is just, I don't have any data to back this up. It's just me walking around soybean fields in northeast Louisiana. The earlier planted fields, we may plant soybeans here in the last week of March. So those earlier planted fields in high yield situations seem to suffer the most.
Okay.
Now, if that's because we get more rainfall earlier in the season, or it's cooler, that fungus may not like hot weather. So that could be a factor too. So definitely there's a lot of environmental factors involved.
Right. So that statement you just made, kind of scares me a little about, it doesn't like the hot weather as we think about it moving more north or being more problematic in some of the big soybean producing states, right?
It's a concern. It's a concern. The folks up north have us beat on acreage for sure. So it's definitely a concern. We don't know. We need to go to those states and see if we can find it, or the specialists in those states we could go up and join up with the specialists up there and go on a hunt for it and see if it's there. If it's not there, we don't have to worry about it. So, but I would think at some point the cooler temperatures might be detrimental. There's some clues in those old Herbaria samples where, they were out of southern Florida, southern Louisiana, the Caribbean. It would make me think that maybe it's kind of like the hot area is better. But when I say hot, I mean super hot, like summertime, July in Louisiana hot, it tends to kind of slow down. So
Right. Well, I like this idea of you coming up and we're all going to go on a hunt together to have
Yeah, I
an
do too. I do too, because
yeah.
it'll probably be hot down here and I want to go somewhere where it's nice and cool.
Well, we'll talk more about that afterwards. I'm going to ask you another couple questions from the Q&A.
Sure.
So have you seen any interactions of taproot decline with other diseases or pathogens?
I don't have any data on it, but it would not surprise me a bit. Rarely, when you go to a field that has soil-borne issues, is it just one. Last year there were several situations where I was looking at taproot decline and southern blight
Mhm.
and trying to figure out which one was doing the heavy lifting. So it's, I would guess yes, but I haven't done any research on that, and that's really a good avenue to go down. And
Right.
it makes you, the thing that pops in my mind is interactions with nematodes.
Okay. So any thoughts on if it might be worse with, some foliar diseases like Cercospora leaf blight or any of those?
I don't know. Anytime a plant has more than one disease, I would say it's definitely worse. I haven't seen any interactions with that and taproot decline that have popped out enough where I wanted to do an experiment and looked at it and look at it. So um, unknown. I'll just say I don't know on that one.
Yeah, no problem. So another question was about primary infection. Can it occur in the reproductive stages or does that infection happen earlier?
It's tough to say, because if you can see symptoms of the entire growing season. And it seems to get worse during pod field. So it's hard to say if there was an initial infection in those plants, or I'm going to personalize soybean plants right now, but they're working really hard to fill those pods, right? So they're taking resources, so that maybe it makes them more susceptible. Or, they were infected earlier and now they're stressed, so now the disease symptoms are showing up. It's hard to say. We don't know. That's definitely something that needs to be worked out as far as the disease epidemiology and the exact mechanisms, the exact host pathogen relationship.
Right. So what about correlations in yield loss and severity among different maturity groups of soybean? So have you seen it worse or better in one maturity group versus another?
I don't have enough data to look at that, but I suspect we probably will eventually if we keep doing this variety screening, the inoculated variety screening, we could probably tease that information out of that data.
Okay. So seed-borne, can it be carried on seed?
That's a good question, and the only evidence we have of that is an isolation of the Xylaria species from soybean seed in 1979. And I think that's an important point. It's a really good question because if you noticed in my presentation, I had a picture of some soybean seed uh, that were infested with Xylaria. I picked those up. Those fell out of the combine the year before. So I
Okay.
just happened to look down and notice they had the fuzzy fingers, or the dead man's fingers, coming out of the soybean. So I thought that was an interesting observation, and it definitely made me question whether or not the organisms in seed. It's just that's more research we need to do. That'd be a pretty straightforward research project for sure.
Right. I think that's, yeah, definitely something to look at. So what about irrigated and non-irrigated soybeans? Any associations with irrigation?
I would say yes on the soils that are drought prone, and I'm speculating here. This is based solely on observations, my observations. Um, it seems to be worse in fields that are irrigated on the Macon Ridge, what we call the Macon Ridge in Louisiana, where we have drought prone soils. In the Delta, not so much, but if you're in an area where you have drought prone soils and you've got non-irrigated versus irrigated, your major issue is irrigation, not taproot decline, if you don't have water.
Yeah. So going back to like my new favorite term, fuzzy fingers. So Would fuzzy fingers be a diagnostic sign if infected soybean plants are incubated in a moist chamber in a diagnostic lab?
Possibly,
Yeah, so
because
could
this fungus will produce those in culture.
okay, so you could maybe, if you were incubating your soybeans and you saw those fuzzy fingers, you could say that's taproot decline.
Yeah.
Okay.
They're usually emanating from soybean debris. They're usually coming up from growing out of soybean debris. So, yeah, I'd say it's possible.
Okay. Good. Well, so I have a couple more types of questions. I was just going to ask some more general questions for you. So I think all of this research, I mean, you've talked about there's so much more research to do because it's a new disease. But why is it important that we have this type of research? You know, you have a broad swath of research, but really, if you can kind of try to articulate why is it important that we do this type of research?
Well, you know, a significant portion of my growers are suffering financial losses from this disease. So that's my underlying motivation. Is you know, it's a problem that they want solved. And in talking with consultants and other stakeholders, you know, I get questions on it all the time. So it's a you know it's a significant disease. It's just like any other significant disease. You want to have the latest research on it. And you take any foliar disease. Frog eye is real popular with people, or unpopular, whichever way you want to look at it. So yeah, if it's affecting my growers' bottom line, that's number one. Number two, it's important to the scientific community. The novel aspect of it. It's new research, so that's always exciting and important. And you don't ever know what's going to come of it. You know, something incredible could come out of new research. So that's why we do it, to answer questions. So
Yeah, well, I think it's support. We have that slide up still about your support, and so I think that sort of, you know, strengthens your argument that this is important. Is that the checkoff dollars are funding this, so obviously the farmers want those answers, right? And
of
so I think
course,
it's important. Yeah, and also I think what you said is that, you know, it's really important for all of us, even as plant pathologists, is that you're doing this work and it helps all of us sort of answer our farmers' questions, and that's really important too.
sure.
So I have a couple more questions from the Q&A. So would you say any of the symptoms of taproot decline are similar to what you see with soybean cyst nematode?
I don't see much soybean cyst nematode, but I would guess yes.
Okay.
I could see where it could be confused with soybean cyst, just based on presentations I've seen from other people.
So,
Anything that the yellows that ends up with yellowing soybeans early, it can be confused with. It went to, I mean, I think I had a slide with 9 different things it could be. I mean, there are a lot of different diseases there that you can confuse with taproot decline.
yeah, I like that slide at the beginning where it had that sort of that long list of lookalike, because I thought that was really telling how easy it is to confuse with other diseases.
Yeah.
So with isolates of Xylaria, do they all produce stromata? And can you culture directly from the stromata, or should you stick with a mycelial transfer?
I would stick with the mycelial transfer. I think Teddy had some success with transferring from stromata, but the best way that I've found to isolate it is surface sterilize affected roots, split the stem, and there's a white cottony mycelial growth right in the pith. And for whatever reason, it usually transfers right from that to any growth medium. I use PDA.
Okay.
Teddy also characterized it on oatmeal agar.
Good. Okay, and I think there was a question about your variety trial and how much it would cost to enter a variety in that variety trial. I think someone's looking for to use your location.
You'll have to talk to our soybean specialist. I can't remember the charge right off the top of my head, but it's Dr. David Mosley, and he's in Alexandria, Louisiana. And I collaborate with him on this trial. He sends me the seed from for his entries for the Louisiana OVT.
Looks like there's some interest in getting some stuff tested in there.
Yeah, I'll plant it.
Yeah,
it keeps getting bigger with Mississippi State and University of Arkansas wanting to add varieties to it. So
yeah,
we may not be able to harvest it that many entries, but we'll do our best.
Yeah. Yeah, well, if there's any other questions, please put them in the Q&A now, but I'll sort of start wrapping it up. And Trey, if you could say, really, if there's one thing that farmers can take away from this talk, what would you want them to remember today?
When you see that foliar symptom, dig deeper, because it may be something else. Could be a lot of different things, and disease identification is key.
No, I think that's really good advice. You know, we try to encourage farmers to dig up, pull those plants, split the stems, but I think also maybe we need to be getting the word out that they need to be looking for those fuzzy fingers too.
Yeah.
Looking.
Call an agent.
Yep.
Call an agent. Call a specialist. Just make sure you have it identified correctly, because if you think it's something else and you're using control measures for that particular disease, it may be the completely wrong approach. So
right.
That's the cornerstone of management is ID.
No, I think this has been great. I've learned a lot, and I think all of the attendees have too. So it's been good to kind of hear it from you about this new disease. And this will be available on the Crop Protection Network website. And so others can watch it as well, and also get certified crop advisor credit for this on the website too.
Yeah, I want to thank everybody again and wish everybody good luck in the coming season.
Well, we appreciate it, Trey. If there's I think there might be one other question, and this one I think is going to be specific to the state. So this person's asking about how farmers need to bring in samples. And just like Trey said, you know, talk to your extension agent or whatever extension entity you have in your state, and they'll help you learn how to bring in those samples.
There's also diagnostic centers. Most universities will have a plant diagnostics lab. Mm-hmm. They might can help as well. You could, you could send me a picture. I might can help. So I don't mind doing it.
That's right. Well, also this, like I said, this webinar recording will be hosted on the Crop Protection Network website. If there's anybody wants to watch it again or get credits for attending today's webinar. So with that, I will put that back to Kayla if she wants to sign us out.
Well, thank you so much to Dr. Trey Price for that wonderful presentation, and to you, Kirsten, for asking all those great questions and moderating a little bit. We really appreciate that. Like you said, really great information that I think a lot of people are going to be able to use. And anytime you have that many questions, you know that you're doing something right. So that's awesome. I just wanted to remind everybody too that there is one more presentation in this Crop Protection Network Spring Webinar Series. That's coming up April 6th at 2 p.m. Eastern, 1 p.m. Central Time, so the same time as today. And Dr. Carl Bradley from the University of Kentucky is going to present Resistance is Futile, the latest on fungicide resistance in soybean pathogens. So you can check that out then. And I put the link in the chat where you can actually go and register for that webinar as well. But thanks again, and thanks for all the great information.
Thank you, Kayla.
Thanks.
Bye, everyone. Have a great day.
Yep, take care.